Yay. All right.
Thank you.
Good.
Mm.
So welcome, everybody. Let's start with a round of introductions. I guess I can start. I'm Steve Glavis. I'm responsible for global sales at Pixelgen. And my background, I'm a molecular biologist, and I've worked with a series of different companies, any of them within the molecular biology space. So 10x Genomics, Thermo Fisher, and its many iterations over the years in all the different techniques that they have. I've also worked in public health, and that was for setting up Public Health Sweden's next-gen sequencing pipeline once upon a time. Annika, why don't you go next?
Sure. Hi, everyone. My name is Annika Branting, and I'm the Chief Commercial Officer here at Pixelgen. So heading sales, marketing support, and also product. We're super excited to have you guys on board and look forward to the collaboration with BioStream and look forward to what we can do together in Japan. This is a very exciting technology that we think a lot of researchers will have great use for. So thank you.
Thank you.
Okay, then maybe I can go. My name is Anna-Lotta Schiller, and I'm responsible for global marketing. I have around 30 years of experience within the biotech field, working mainly in commercial, both towards distributors and as a part of a distributor organization, startup organizations and big companies like GE Healthcare. So it's a wide range going from everywhere from genomics to proteomics. And I'm looking really forward to working with you and supporting you with marketing material and marketing activities. Nice to have you on board.
Yeah, nice to meet you.
All right, so I can go next from the Pixelgen side. My name is Louise, and I am the field application scientist here at Pixelgen. Been with the company now for almost 2 years, and I was previously in R&D, so I've done a lot of R&D work as well, and really working closely with the technology. But now I'm on the commercial side and with the application part of it and supporting there. So we're super happy to have you on board, like everyone else said, and I look forward to helping you with all of the support material for the application part.
Okay. Thank you.
You're muted, Stefan.
Yeah, sorry about that. Yeah, I'm Stefan Petkov, and I am a field bioinformatics scientist at Pixelgen. I have a PhD in vaccinology, immunology, where I spent a lot of time analyzing a lot of genomic and proteomic data. So yeah, I'm looking forward to working with your team as well and helping you, get the analysis sorted out and handle any issues that pertain to that.
Okay, thank you. Yeah, so from BioStream side, I'm a now Hosono of application scientist of BioStream, and yeah, I have more than 20 years of experience as an application scientist, and my career started with the Applied Biosystems first, responsible for DNA sequencer, and then I moved to Riken Research Institute as an application scientist, research scientist, and then I moved to a Fluidime, and yeah, and I worked for them for 6 years. After that, I joined BioStream. Yeah, and now I work for BioStream. I have worked for BioStream for 6 years, and now I'm responsible for a single cell RNA-seq product and the special proteomics and the special genomics product. Yeah, yeah. I'm really excited about your technology. Yeah, thank you so much for giving us valuable opportunity today. Yeah. And Matsumoto-san.
Hello. Hello, this is Seishi Matsumoto. I'm a sales rep for West of Japan, but not a scientist. And I'm sorry, I'm on the way from customer side to my home. So I would like to join this meeting with a mute.
Okay. No problem.
Sorry.
Nice to meet you.
Sorry.
じゃあ、清水さん、いいですか？
My name is Hiroyuki Shimizu. My English is very bad. Please slow English, please. I'm sales side for East Japan. かな？
うん。
そんなもんですね。はい。
はい。
Thank you.
Thank you.
Yeah, thank you so much.
Okay, so let's go on to the next point.
Yep.
So expectations are what we'd like to discuss now. Would we like to start from your side and hear what you have to say about your expectations and your experience working with other companies as a distributor? That'd be very helpful for us.
Okay. So now actually our BioStream is now focusing on the single cell related product like BD Labs, Vector Dickinson's BD Rhapsody and the Scale Bioscience. Scale Bioscience is a single cell analytic company using a combinatorial barcoding, and we also contact with Curio Bioscience. They released the product this February. Is a Curio Seeker. It's based on ThreadSeq. Developed by Broad Institute. Yeah, and we also selling Acquire Bioscience PhenoCycler fusion system. Yeah, and we have a lot of customers in this single cell related regions, and now we sold the BD Rhapsody more than 70 instruments in Japan, and we also sold 30 instruments of PhenoCycler Open and PhenoCycler Fusions. Yeah, and now we just start to sell the Curio Seeker ThreadSeq product, and we have already 40 prospects. Yeah, it just started selling since this August, and the promotion is really successful, and we now have a lot of prospects. And as you know, in Japan, the fiscal year is start April and the end March. So the January to March is a very important time for us, because many people have a remaining budget. So it's good timing to start promote your product. Yeah.
That's good.
So probably Yamato-san is some preparation for about the expectation for you. So I think he's joining in a couple of minutes. So yeah, after Yamato-san joining, we continue to talk about this topic. Is it okay?
Okay.
Yes,
Okay.
that is okay.
Yeah. Okay, thank you.
Okay, so from our side, the things that we'd like to make sure that we provide you is all the support you need. Louise is going to be the one that can help you most with the support side. Both wet lab, she'll be your main point of contact. And any questions you have regarding support, either from the customer or internally, Louise will be happy to help you with this. And I think one of the most important things is that we have a continued communication channel.
Yeah,
it can be written in Japanese because Google Translate works very well these days. So don't be too concerned.
Yeah, you're right.
So we'll do what we can to make sure we understand each other, and we'll do what we can to make sure we gather information from your customers so we can help support them as best we can.
Mhm.
So these types of things are what we'd like to do. And then we have Stefan on our side who can do quite a lot from the bioinformatics support side.
Mm.
And of course, both of them have access to our R&D departments, so we can ask the more detailed questions.
Yeah.
We also have somebody that is not on this call today, and that is Hanna, and she's in charge of applications support.
Mhm.
And application support today is basically optimizing different types of sample preps at the moment. And then we start moving into different types of functional tests with biological outcome. So we've already been in discussion a little bit with Yutaka Suzuki
Yeah.
about some aspects of sample prep, fresh versus frozen. We've also started talking about maybe we could work with them on developing an application note, so that could describe the fresh versus frozen sample.
Okay. Yeah. It's a very good story for us because he is a very, very strong influencer in Japan. Yeah.
Yes.
So
that's
everyone knows him.
that's the main reason why we wanted to have an application note with him, so we can help you be successful.
Yeah. Thank you. Yeah, so much.
Okay. Should we go through the presentation a little? Just touch on a few points to make sure everyone understands the technology. I'll skip the applications and mostly focus on the functionality of molecular pixelation.
Yeah. Probably the Shimizu and Matsumoto-san is not familiar with your technology in detail. So could you please explain that?
Yes.
Yeah.
And also, please view this as an introduction. We will spend more time on this, have more sessions. So, don't feel it's a little bit overwhelming sometimes to hear about our technology for the first time. So, we'll make sure that we talk about this more. And this is, the first introduction.
Okay. Yeah. Please.
So if you have any questions during the presentation or after the presentation, please let me know. I'll send you this presentation as well, so you can review it at your own pace.
Yep.
I'll send it via Anna Lotta's. She's setting up a particular channel, place to a repository, so you can get access to all the documents. Have one place to go to receive everything.
Okay.
So it'll make it easier than sending emails all the time.
Yeah.
All right. Let's go to the technology here. I will start with specifically technology, skip everything else. What we're doing is we take something that we call a molecular pixel. Essentially what that is a little bit of DNA that we have amplified versus via rolling circle amplification. And what we've done with this is that we've input what we call a UPI. That's a barcode. There's a unique barcode for many, many templates. So we have billions of unique molecules. So there are billions of barcodes. And all these barcodes, we then amplify with rolling circle amplification. So it becomes a long string of many of these barcode regions. So we have many barcode regions per unique molecule for these molecular pixels, and we have billions of them. We use this as a reagent in our assay, and it is the core technology in our functional assay. This makes our product very unique in the sense that we are able to capture many hybridization events on a cell surface. The workflow, despite sounding very complicated with the technology, is very easy. It's a simple workflow with 4 steps, and then you input it into a next-gen sequencer. The first step is to fix the cells with a standard chemical called PFA. Many labs that work with flow cytometry, they work with PFA and are familiar with this process. The same thing can be said of antibody oligo conjugate binding antibodies. So this is what we stain our cells with. These particular staining processes are very similar to what people experience in flow cytometry labs. So again, people who work with flow cytometry will be familiar with this process. We recommend in the first step around 500,000 cells. In the second step, we then decrease the amount of cells again to around 200,000 cells. And then we enter into our unique assay, which is molecular pixelation. And it is a two-step protocol. And the protocol allows us to use our rolling circle amplified product, our molecular pixel, with the unique barcode to capture antibodies in one particular region. This creates a neighborhood. This neighborhood only has a few captured antibodies. These antibodies all have an oligo tag on them, which then is extended at each of these steps. And each of these steps introduces a barcode. So in this first step, you have a neighborhood created of antibodies. In this third step, you have a neighborhood created, this yellow circle, of neighborhoods of antibodies. So it's a multi-layer barcoding. So this one tells you where the neighborhoods are located. This one tells you where the antibodies are located. And this one tells you the identity and abundance of the antibodies.
Steve,
So
can I ask a question about the size of neighborhood? What is the size of the DNA pixel in average?
Yeah, so the molecular pixel, we've measured it with electron microscopy. That was on the previous slide. It is roughly 100 nanometers in size.
100 nanometer.
However, we don't know how much it spreads out when it captures and creates a neighborhood. It could be a little bit larger, but we don't know for certain.
Okay, thank you.
So to make this very simple, our assay is able to capture the data that you commonly see on flow cytometry. Also, you see the spatial information you capture with microscopy. So you combine both the abundance data you gather from flow cytometry and the spatial information you get with microscopy without imaging and without instrumentation. So this whole workflow is done without any instrumentation whatsoever. It is standard laboratory equipment like thermocyclers, pipetters, and centrifuges. In detail, the structure of our antibodies looks as such. We have an oligo that is conjugated to the antibody. There's a barcode and a UMI for each individual antibody. And that gives us an identification of what antibody this is. It lets us count the number of antibodies as well, since it has a unique molecular identifier. On this oligo, you also have a hybridization region for the Pixel A rolling circle amplified product. And it hybridizes here. And at this point, you can introduce this barcode called UPIA, where there are billions of unique molecular barcodes, which allow you to gap fill by having a primer and some enzymatic reactions to extend this particular oligo that is connected to the antibody. This also is hybridizing to other antibodies in the neighborhood, as you see here, here, and here. We then enter into the next step. It is identical to the pixelation A step. However, it has a different hybridization region for the end of this molecule that is extended on the antibody. You then introduce the unique pixel identifier B oligo barcode, again through the same means of a primer and a gap fill reaction. You then have a very long oligo that is attached to this antibody. These 3 barcodes give you the neighborhood, the neighborhood of antibodies, and the unique identifier for the individual antibody and its abundance. In the following step, you do a PCR, and you then add on Illumina adapters. This allows you to have sample indexing, and it becomes compatible with Illumina sequencing. We have tested this with the NextSeq 2000, and we also have customers using the larger, newer NovaSeq, the Plus systems and the X systems.
Can I ask one question about the number of reads needed per sample? Yeah, it's very important in terms of cost. Yeah, so what is the recommendation? Yeah,
we recommend 120,000 reads per cell.
120,000.
Yes, I have it on this slide here.
Okay, yeah, thank you.
120,000 reads per cell.
Per cell. Okay.
Yes.
I see. Yeah.
And then we have a trimmed read length that we recommend. It can be any format. So if your customer is running exome sequencing or something else on their system, you can also run this in the same run. So we have software which will trim this down to this length. So the sequencing does not have to be this, but this is what we recommend. So you can do 2 by 150 as well, and that is acceptable.
Yeah, the number is a little larger than the read in single cell RNA-seq, right? And the in single cell RNA-seq is a few thousand reads is sufficient. Yeah, so the single cell RNA-seq customers maybe feel a little larger reads needed. Yeah.
Yes. So single cell sequencing generally recommends 30 to 50,000 reads per cell. But from what I've heard from customers that have used both our system and single cell RNA-seq, they find that the data produced by single cell RNA-seq is, they call it disperse. So it isn't very dense. Ours is a very rich data set, which is why we recommend so many reads per cell.
Yeah, okay. And the I think a few NGS newcomers exist in United States now, like Element Bioscience or something. Yeah. So do you plan to develop the protocols for that?
At this time, we are not. However, we don't see an issue with it being compatible. The most important thing is that when you make the library, is that it's compatible with the sequencer of choice. For now, we support Illumina sequencers, starting from as small as the NextSeq 2000.
Yeah. Okay. The Illumina sequencer is the most expensive. So.
Yes.
Yeah.
Yes, we know, but it's also the most validated system.
Okay.
Good. Would you like to see the concept of how we create the neighborhoods, or is this enough for the initial call?
Yes, please.
Okay. So after we have made these neighborhoods with molecular tagging and introducing these 3 barcodes, we then try and group them in neighborhoods. So this is a representation of a cell. And in this particular example, we take one antibody, which has a tag, and then we stain it on the cell. And the next step, in the first pixelation step, is we capture a few antibodies in one neighborhood with a molecular pixel A. This molecular pixel A, you then put its barcode in. So in this case, it is a dark gray color. The neighboring neighborhood, which has captured 4 antibodies, has its own unique barcode, which is represented by an orange color here. And the same can be said of this white neighborhood below. Then in the following step, we introduce the molecular pixelation B barcode. And this is represented by different colors. Here it's a magenta, here it's red, there it's yellow, there it's blue. But then you see that the subpopulation also has the pixel A barcodes, which are different. So it overlaps these previous neighborhoods. So it's a way to make a very tight network of the entire cell surface for this one antibody in question.
Yeah, okay. So is this 2 barcode stories sufficient for defining neighborhood?
Yes, so these barcodes are required to create a neighborhood. So this is a neighborhood of neighborhoods, and this is a neighborhood of antibodies.
I feel it's more barcodes, the more exact cell neighborhood definition, right? But
Yes. With these unique barcodes, we have so many. There are billions of these barcodes that allows you to identify very unique neighborhoods.
Okay.
And we have only done 2 molecular pixelation assays in this particular experiment. So you have the pixel A and the pixel B. And this is required to have this capture of antibodies in the neighborhood and then neighborhoods of neighborhoods. You can even do a 3rd one and whatnot, but that's speculation. We don't have an application for that at the moment.
Okay.
So after you read this on the Illumina sequencer, you have what's called an edge list. And it's made up of the barcode for the antibody, the barcode for the pixel A, the barcode for the pixel B. And these are represented by colors. So as you make a graph, you then find this individual color here, and you want to connect it to the pixel A barcode. So these small circles represent the pixel B barcodes, which we referred to in a graph setting as a node. And you want to go to the next node. Between the nodes is what we call an edge. This edge is where the antibody lies. So the event that occurred is the antibody binding to the cell surface. And that is represented by this edge. So that binding to the cell surface occurs somewhere on this line. So we start with this edge list, and we say this color is connected to that color. You can also see that it's connected to this color because these 2 colors are the same. So if you look, you see this one is connected here as well as here. But you can also follow down this line to see which one is connected to which. And what you end up doing is you can follow these colors. Here's the yellow one. There's the yellow one connected to this grayish blue. That's here. And it's also connected to these 2 colors as well. And that is over here and here. But that allows you to recreate an overlay of an entire cell. So again, here's this data analysis slide where we have the output, what recommendations are for sequencing. You can also see that we have different reports that you can work with. This is where Stefan will start helping you. How do you work with Python and R as our output? So we provide a software package which allows you to have all of these different things. It's a standard package. It's online on our web page. Louise and Stefan will be happy to show you exactly where all these resources are on our web page.
Yeah.
But what ends up coming out is you can get a visualization of this, and that's represented by this forced spherical representation of a cell. And here you see a dispersed surface of the one molecule, but then you can choose looking at the polarization of a particular event. And polarization is a term we use for saying these markers occur on one end of the cell.
Yeah, that's great. Do you plan to collaborate with the data analysis scientists, like a New Genome Center, Satija Lab or something? Because I feel it's the target customer is a single cell customer, and the student is very familiar in single cell analytic researchers. So the probably the customers would like to analyze your data with single cell analytic data or other single cell related data, right?
Yes, so we're finding that there's an interesting mix of customers that find this interesting. So we gave a seminar yesterday at one of our large sites in Sweden, and what we expected was many more single-cell RNA-seq customers, but it was more often the people who worked with imaging. So they want to see where the surface proteins are located on cells. They want to see how they interact with one another and how they combine to start signaling. So we saw that quite often yesterday at our presentation.
Okay.
It is a very new customer segment.
May
I just add to that, because Hosono-san, I think we also thought the same, that the single cell customers would automatically understand, and they would be naturally the early adopters. But also what we see is that there's a segment in the single cell customer segment who doesn't really understand proteins. They worked so much with transcriptomics that they have almost like a blind spot for proteins. They don't understand the value. And so I think that's a challenge that Pixelgen needs to work on, with messaging and education for this customer group. What you're saying, when we can start comparing the transcriptomic data for single cell to the pixelation data, we can show the added benefit also with pixelation data. Then I think the entire group of the single cell community will become interested. But a little bit surprisingly to us, it hasn't come as natural as we thought. And we think it's this disconnect between understanding transcriptomics and understanding proteomics.
I agree with you. And additionally, recently the single cell customers start to the experiments of spatial biology, right? And I think almost everyone of single cell analytic customer interested in spatial biology, and the KOLs would like to see their cells in a special format of more higher resolution. And as you know, it's the nanostring cosmics and the thanks Xenium is a very, very high resolution, and which can see the molecule to molecule connection. I think the many people is pay attention to the molecule connection interaction in the molecular level. So so I feel we our customer really overlap with your customers. So it's my impression.
Good. Should we go on to the next topic? Is discussing marketing resources, where those are available, and we'll give you access. Analata, would you like to show where the resources will be found?
Yeah, and I will go through. So I will just share my screen as well. So can you see my screen?
Yes.
So I went into your website yesterday to find out a little bit of what material would you seem to prefer. So please correct me if I'm missing something. And I put together a folder where I have included a short company description for you. And I also included the company brochure and the product sheet. Then I have a separate folder within that you can actually then have the company logo, and I didn't know what type of file you would prefer. So either a PNG file, JPG, or EPS files are included there in different formats. And of course, the protein list and the animation and illustrations that could be useful for you. And also the article that is waiting for publication at the moment. And I don't know if you're interested in the coloring of what we use, but I put in the hex code just for your needs. And in regards of product sheet and so forth, I don't know if you're interested in getting these translated.
Yeah.
Yes.
This is Ivaguchi. I'm sorry, attending so late. So we've almost finished the translation based on product sheet that we download from your website. But
okay, so this one here.
no, it looks like a little different. That is a newest version?
Yeah, we have two different versions. We have one that is printable and one that is more easy to read online.
I see.
So I think that, and do you want access to the pictures?
Of course, if we can do that.
Yeah, so I will be happy to assist you in regards to that. And also, so I have included a folder for you, which I will give access to all of you that were on the team and any additional person that from your company that you would see fit to need the access for this.
Okay, I will control that. Okay, thank you.
Yeah, and then in regards of because I know that we are planning on a press release, and in my work in setting up that, in preparing for everything and be up and running, it would be very nice if I have the possibility of some logo files from you and a short company description, as well as the necessary contact information for sales and support, and of course the web link, so everything is correct coming from you.
Okay, so I can easily to generate.
Yeah.
That
And will there be a specific person that I will communicate when in regards of marketing activities and stuff when we will have webinars or do joint?
Okay, at the moment, please, I'm looking for the real marketing guy, but at the moment, please contact myself and Hosono.
Okay, I will do that. And please let me know any other material or information you would need in regards to marketing.
OK。そう、細野さんね。
うん
なんか今使ってるようなプレゼンの資料っていうのは必要じゃない
必要ですね、必要です。
うん
で、一応、後からくれるって話なんですけど。
あ、本当わかった。
So, yeah, we actually sometimes make a webinar presentation video and upload to YouTube. And yeah, so if possible, I would like to do that. So could you please give us some presentation file or
Yeah.
Yeah.
And we will collect everything like that in the folder.
Yeah.
And please let me know that you have access to it, so no problem with it.
Okay.
Great.
Yes.
That was
So-
all on my side. If you don't have any additional questions, so otherwise back to Steve.
Yes, so let's continue on to the plans for support, both from the wet lab and bioinformatics. Louise, would you like to introduce what we have for tools available for support?
Yeah, definitely. I think that we could also set up a meeting for this to discuss more specifically and also go into detail with the protocol and so. But I can share, let's see here. So, can you see my screen?
Yes.
Yes.
Let's see.
Now it's gone.
Okay. You see my screen now?
Nope. Oh, coming.
Coming. Okay.
Yes. Now I see it again.
Okay, great. So I think that we'll set up a meeting to discuss, as I mentioned, more in details with the protocol, how to run it, and how everything works, and what the standard procedure for getting people started looks like. And I can also put this in the same folder that Anna-Lotta had, or we make another folder. But if you go to resources here on our Pixelgen website and just scroll to the bottom, you have also the user manual. So this is always going to be the mostly updated user manual. Then you can just press that and that will be downloaded. So I think that's what the material is, apart from these presentations that I tend to show to customers. So what I do is usually I perform like a technology introduction, which is quite similar to what Steve did now, but it's maybe a bit more detailed towards the technology. And then another session after the customer has read through the user manual. We take a session to deep dive into the protocol and the workflow, and also discuss the user manual. So going through the different steps there. That's usually how I do it. And then there's another list that's not on the site here that I will also share with you. That's called Getting Started. And maybe I can share this tab instead. So it looks like this. Yes, you can see that one. Great. So it's a getting started checklist, just so that customers can see that they have the experimental design and the cells ready for running our technology, having the kits available, having the times to run it, and the list goes on of just a super easy checklist for them. And then there's also included here with what equipment they need to have. And as Steve mentioned, you don't need any specialized equipment, but of course you need a centrifuge and a thermocycler and standard lab equipment. So that's also available here. But I can do a similar manner that Anna-Lotta has done with, instead of having a marketing folder, I will do a support folder for you and share it as well.
Okay.
Maybe we could have a link to each other in that, Luis, so that it sticks together easily for this distributor.
I think we could do a main folder that's just called Pixelgen, and then we can divide it in there.
Yeah, or BioStream.
Yeah, my Pixelgen BioStream. Yeah, sure. And I will, but as I mentioned, let's set up another meeting as well to go through more of the support is handled and the onboarding for customers.
Okay. Lisa, do you usually give a customer training for the first time customer? No.
So there is no training required on site. It works very well to do it remotely. That's how I've been doing it so far, and it's been working well. But it's important that all of those different presentations and all of those different documents that I typically send out are sent out, so that they have all the material, and so you have that active discussion.
Okay, thank you.
Yeah, of course.
I can also go through the data analysis materials that we have.
Please, yeah.
I'll also share a screen here. Is that loaded?
Yes.
Yes.
It's nicely.
Yep.
Right. So we have an extensive set of resources on our website. Location is software.pixelgen.com, and it can also be accessed through the main site, pixelgen.com as well. But here we've collected everything that is basically needed to run through the software. So we have Pixelator, our open source product that processes sequencing data and gives you the data for downstream analysis. And we have documented how it works, all the separate steps in analysis, and algorithms that are kind of involved into the analysis. So that's some theory here as well. Then we also have an infrastructural version of it, which is based on the NF4 workflow orchestrator, and that is for running high number of samples on a high compute cluster. And that is also kind of documented here. There's extensive explanations on how it works and how to get started with that. Furthermore, we have a extensive set of tutorials on how to work with the data that has already been processed by Pixelator. So we have everything from getting your Python or R environment set up, and also getting started with basic data handling, quality control samples, annotation, and all the usual steps that are involved in. It's also part of single cell analysis, but those are documented here. We also have some advanced topics that can sort of guide you through 3D, creating 3D visualizations with both R and Python. So those are available here, and then detailed kind of manuals on how to produce those 3D visualizations. And lastly, we also have a few curated data sets that we've included for everybody to explore and kind of get a little bit of hands-on usage of the tutorials. So we have data sets that are downloadable. You have the raw data. You also have the output of Pixelator here. That is making it easier, so you don't have to necessarily go through the raw data. You can just go down to the actual analysis. And then we also have sample reports here that are available for kind of viewing. I think that is it. But we'll of course collect all these informations along with everything else. But it would be nice to also have a contact on the BioStream side that we can communicate, and basically also set up a more kind of detailed introduction and how to go through all these things.
Okay. Stefan, could you go back to previous screen and software?
Yep.
Yeah. And that Pixelradar and NF Core software needs a high-spec computer. What's the spec of the computer for that?
I mean, it is basically a high compute cluster, so it would be a server on the premises of the laboratory. It can be a core facility. So it is flexible. It depends on what resources are available. You can of course also run that. You can run Pixelator on a single computer, but I mean that will be kind of very limiting, as you can imagine.
Okay. This software is based on Windows or Mac or Linux or something?
It's platform agnostic, so there's no requirements for any particular environment in terms of operating system.
Okay. Thank you.
Yep.
Good. Okay, I think now we need to plan how often do we want to meet and communicate between us. I would suggest that Louise and Hosono decide what works best. I believe you can do that via email and set up a schedule that works well.
Okay.
Iwabuchi and I think we can plan how we want to work with the cadence of sales reporting and communication of leads. Analotta, who would you like to communicate with specifically in marketing? Is it Iwabuchi as well?
I think it was Hoso-san and Iwabuchi as well. I think
both, yeah.
Wasn't that correct? Yeah.
Good. So why don't we plan, would another follow-up meeting in about 1 week's time be acceptable, or is it a little bit tight for the end of the year?
So seriously, now is close to the middle of December. It's very tight. So maybe next one is 細野さん、クリスマスの前後くらいできそう？
そうですね。はい。
Okay, next one is the before and after the Christmas. What do you think?
We'll do after Christmas.
Okay, after Christmas. That
And
means
then
that. Go ahead.
No, then we can just set up this also the deep dive into the technology and protocol.
Yeah.
And maybe meanwhile, we can exchange the material needed so that we can get our websites updated. But that we might just do by email easily, I think.
And Iwabuchi-san, I will send you the press release that we've been working on. And just for you to say that you think it's okay.
Okay.
And when you approved it, we will send it out.
Okay, thanks a lot.
Thank you.
I appreciate your
And then one point that it's a tactical thing. We received a lead from the Japanese region, and I wanted to know how would you like to handle leads? Should I hand them directly to you, or would you like me to follow up with them?
No, could you send us the lead information?
Yes,
first?
I'll do that.
That is pretty important. And as it depends upon request from the lead, so maybe we need to contact you.
Yes, perfectly fine.
Okay.
Yeah, we're always happy to hear from you.
Did you get? Oh, sorry. Did you get any lead already?
Yes, we did.
You sure?
Yes.
Wow.
From Riken Genesis. It's a service provider.
They contacted us before, exactly.
Yes. So I had a feeling you knew them already.
Who is this? Kondo?
I'd have to double check the name, but let me see if I can find that again real quick. Eriko Matsura.
No, I have no idea. Maybe technician.
I will forward it to you right now.
Okay, thank you.
エリコ松浦。OK
いますよ、その人。
ん
いますよ、その松浦さんって。
知ってる？
メールでコンタクトしたことあります。
Our sales rep knows her.
Oh, good.
Okay.
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 Thank you very much.
Thank you so much.
We greatly appreciate your time and definitely are looking forward to working together.
Yeah.
Yeah, definitely. Thank you for your time also.
Thank
Thank you so
you.
much.
Yeah.
Thank you.
We're expecting to get a good result as soon as possible.
Yep.
Okay.
As are we.
All right. All right.
Take care.
Okay.
Thank you very much.
Thank you so much.
Bye-bye.
Have a good day.
Thank you.
Thank you.
Have a good day.
Bye.
Have a good day.
Mm.